Okay. Hello everyone. Welcome to today's Global IMMUNOTALK. It's my great pleasure to have today's speaker, Dr. Leng Siew Yeap. Dr. Yeap received her PhD from University of Cambridge, where she studied epigenetic modifiers in maintaining potency of mouse embryonic stem cells. Then she moved to Harvard, conducting postdoctoral research with Fred al, where she developed a novel passenger immunoglobulin allele mouse model system to study sequence intrinsic mutational patterns of antibody somatic hypermutation. And I think that's also part of her lab now. She has been a principal investigator at the Shanghai Institute of Immunology at Shanghai Jiaotong University School of Medicine since 2016. And that's broadly part of her topic in her own independent lab, trying to understand the mechanism that generate real mutational outcomes during antibody somatic hypermutation process, such as indels and multiple base substitution, how the mutation trajectory during antibody evolution contribute to antiviral broadly neutralizing antibody activity or autoimmune diseases. And I believe she will tell us about decoding the antibody mutation trajectory, physiological and disease state. But before that, as our tradition for the Global IMMUNOTALK, we'll ask her a question that she would like to share with our audience. So Dr. Yeap, I think you would like to tell us about how has technology changed your approach to research over the years, and where do you see it heading?
Hello, hi. Thank you for the invitation to speak at the Global IMMUNOTALK, first of all. And regarding the question, I think it's very interesting. My own research, I think the sequencing technology and also the CRISPR-Cas9 technology have definitely improved our understanding of antibody somatic mutation, and also the way how we make a lot of all these mouse models to dissect the mechanisms in generating diversity. And as I present later my talk, I would also incorporate that into the talk. And also, I think towards the future, I think AI would be something that not only useful for us, for our research, but also I think for all biological studies. So I look forward to, to see how AI can change our understanding of antibody diversity. Yeah.
Great. Thank you for that note. So why not please start to share your presentation, where I will let the audience know that it's our new tradition at the Global IMMUNOTALK, that there is not going to be a live question answer session, but you can go on YouTube where all the talks will be posted, and you can post your question at the YouTube, and the speaker will answer. I think at the end of Dr. Yeap's talk, there will be a slide telling you exactly how to use the YouTube channel to ask your questions. Okay, now the podium is yours. So please, Dr. Yeap.
Okay, thank you. Hi, could you see my PPT and my pointer?
Yes.
Yeah, okay, great. So thank you again for allowing me to talk on this Global IMMUNOTALK. I think it's very exciting right now for my lab. We are interested in understanding antibody diversity and humoral immunity. And today I'm going to talk about some of the work that we've been doing for the past few years, and some of the work that we are currently doing as well. So we know that antibody is the secreted form of B-cell receptor. And in the case of adaptive immunity, we know that B-cell receptor and T-cell receptor diversity is the hallmark. And we know that, for example, B-cell receptor, it has a large repertoire from 10 to the power of 12 to 15. And the reason for that is because it allows us to recognize the many different antigens or pathogens that we see daily in our life. So, and also the diversity of the B cell receptor allows the B cell to recognize these antigens with high specificity, and also it generates memory to the B cells. So if you look at the diversity of the antibody or the immunoglobulin, it mainly lies at the N-terminus, which is the antigen recognition region. This region is also called the variable region. And in the variable region, we know that there is this complementarity determining region, which is the region that is most hypervariable, and also the framework region, which is responsible for the structure, and the diversity is less compared to the CDRs. The constant region in the N-terminus of the immunoglobulin is responsible for the effector function. And we know that from works by many labs till now, that there is actually two steps in generating diversity in the variable region exon. And the first step occurs during B cell development. And this process is called VDJ recombination. And during VDJ recombination, RAG endonuclease would recombine the different V, D, and J segments, which lie across 2 megabase along the IGH locus, to generate a variable region exon. In the light chain, the V and J segments be recombined. And this process would actually generate the primary antibody repertoire, which has no affinity for antigen. And most of these antibodies are actually IgM antibodies. And when B cells meet a cognate antigen in the periphery, another process, which is called somatic hypermutation, which is mediated by AID, would introduce point mutations in a variable region exon. And also AID would result in cartridge recombination to switch the constant region, for example, from IgM to IgG, switching the isotype of the antibody. And these processes actually would result in the secondary antibody repertoire, which has high affinity, and would also result in a final diverse 10 to the power of 12 B cell receptors in the repertoire. And B cells at this stage the somatic hypermutation occurs mainly in the germinal center B cells. And through many rounds of mutation and selection, the high affinity B cells receptors would be selected. And at this stage, the B cells can differentiate into memory B cells or plasma cells, which secrete the antibody. And as we know, the somatic hypermutation mainly occurs in the CDR regions. So back in the 1970s, when hybridoma has been, the technology of hybridoma has been identified, David Baltimore and Leroy Hood in their labs, they sequenced hybridomas that generate antibodies against specific proteins. And what they found was that there is a variation in the variable region of these immunoglobulin genes. And in 1982, again, David Baltimore found that there is a somatic variance which clustered in the CDRs of the kappa light chain. And in the discussion, he discussed that there are two alternative possibilities for the clustering of somatic events. Here, I'm just reading this from the discussion in the 1982 Nature paper. It says that it's either there could be a predisposition for mutations in the CDRs, or selection could be operating. And preferential somatic mutation in the CDRs would require a remarkably specific mechanism, but it may occur. So basically, there is two hypotheses for bias of mutations in the CDRs. It's either that the sequence are predisposed for mutations, or there could be a selection. So nobody really knows the answer for this question. And in the 1990s, work from several labs have shown that there is probably an indication of predisposition of somatic hypermutation in the CDRs. So work from Michael Neuberger's lab showed that when they knock in the immunoglobulin transgene into the mice, they could see that mutations are clustered in the CDR regions. But these experiments actually, the results could reflect the integration site of the transgene and also the copy number of the transgene in the mice. Another experiment which look at non-productive alleles by Lipsky et al. They also found that the CDRs are highly clustered for hypermutations. But we do not know whether these non-productive alleles have undergone selection before they become non-productive. So these experiments do indicate that there is predisposition of somatic hypermutation in the CDRs, but there is it is not an actual experiment results that we could be sure of this predisposition of somatic hypermutation in CDRs. And if you look at the textbook, we know that the CDRs are always shown to have more mutations than the framework regions during the immune response. Mutations would accumulate more in the CDRs. So one of the main challenges in studying somatic hypermutation is that we know that each B cell expresses a single B cell receptor. And there is heterogeneity of CDR3 sequences. For example, CDR3 sequences is generated by the combination of V, D, and J segments. And also there is numerous V gene segments in the human, for example, 30 to 40 V gene segments. So it's really heterogeneous and diverse. And also there is a positive selection during the process of affinity maturation. Therefore, if we sequence a B cell receptor, we only see mutations that are positively selected. So this actually results in the inability to measure precise mutation frequencies. And also we are unable to capture the sequence mutation pattern. So when I started my postdoc in Fredolf's lab, we were able to generate a passenger allele mouse model to address the question of heterogeneity of the V gene sequence. So in this mouse model, we could knock in a sequence, any specific sequence in place of the BDJ exon. We call it the passenger allele because it carries a stop codon, which is being transcribed but not translated into a functional protein. And because this passenger allele is targeted by AID for somatic hypermutation, we could capture unselected mutations because this allele is non-functional. And the other allele, it could be a wild-type VDJ rearranged allele, or it could be a knock-in of a productive VDJ exon. So on these productive allele, the mutations would be selected. So there is a difference between the passenger and the productive allele mutations. So at that time, actually, high throughput sequencing was available. When I started, we were just still using Sanger sequencing to sequence all these mutations. But when high throughput sequencing was introduced, we immediately used this technology to look at the mutation patterns of the passenger and productive allele. So here we have two alleles with the same exact sequence, the mouse B18 sequence. This was actually the same sequence that was used or was named by David Baltimore in his 1981 paper. So we knocked in these sequence, one in the productive allele and one in the passenger allele. And what we saw was that in different mice, in 6 different mice, we saw that the mutation pattern of these sequences in the productive and passenger allele, they are very similar to each other. And what we notice also is that the CDRs target high somatic hypermutations, which suggests that actually there is a predisposition for mutations in the CDRs. So how do we know actually these two these two allele, one is productive and one is passenger? So actually we found that in the passenger allele, there are high levels of deletions, which are normally negatively selected because these deletions are normally deleterious to the function of the B cell receptors. So we know that what we are sequencing is actually accurately reflecting either the productive or the passenger allele. So we actually wonder what is the mechanisms that target high somatic mutation in the CDRs. So if you look at the enzyme that generates the somatic hypermutation, AID. So AID stands for activation-induced cytidine deaminase. It's part of the AID ApoBac superfamily. It's actually a single-stranded DNA deaminase. It deaminates cytosine to uracil. And at the genomic locus, it actually prefers the cytosine in the context of WRC, where W stands for A or T, and R stands for A or G. So this is the microscale motif that AID preferred. But if you look at the CDR sequences, it actually do contain high density of WRC motifs. And also this AGCT motifs is the palindromic motif that AID most preferred. So if you look at the AGCT motif in the CDRs, they are more highly targeted than the same motifs compared that is located in the framework regions. So we actually think maybe the sequence surrounding the CDRs could promote AID targeting to these AGCT or WRC motifs in the CDRs. And this, we call this mesoscale sequence, which differs from microscale, as mesoscale would be around 5 to 50 base pair surrounding these microscale motif. So to test this hypothesis, we use passenger allele mice, and at that time, CRISPR technology is available, and we use CRISPR and sgRNA targeting the CDR3 of the passenger allele. We inject these guide RNAs into the zygotes from the passenger mice. And with these experiments, we can get a batch of passenger allele mice that have sequence alterations in the CDR3 due to error-prone repair. And from the 20 passenger mice, 11 of them do have sequence alterations around the CDR3s. And we measure the mutation frequencies of these AGCT from germinal-central cells and use the CDR1 as a control. And what we found was that some of the passenger alleles that have sequence alterations close to the AGCT do have mutation frequency changes. For example, the blue ones, the 5 of them, have reduced mutation frequencies at the AGCT, whereas one of them have increased mutation frequency at the AGCT. And this actually suggests that mesoscale sequence do determine the mutability of this AGCT in the CDR. But at this stage, we still do not know what is the feature of this DNA sequence. So we then collaborate with Fei Long. So what we did was to use a single-strand DNA substrate and a purified AID to do in vitro AID deamination assay. So what we found was very exciting to us because we found that in vitro, CDR sequences are still more highly mutable compared to the same motifs in the framework, which mimics what we saw in vivo. So this is the correlation of the in vivo and in vitro mutation of each of these positions, C positions. The only exception is that in vitro there is no A or T mutations because A, T mutations are generated by error-prone polymerase during DNA repair. So this is very encouraging to us. And when we look at the substrate that AID prefers, we found that flexible substrate, poly-DT substrate, would be more preferred by AID, whereas a poly-DA substrate, which is more rigid, is less preferred by AID. And so this is the molecular stimulation experiments performed by our collaborator, Lin Da. So we found that the flexible substrate actually bind better to the positively charged patch of the AID. So with these experiments, with the biochemical experiments, we were able to pinpoint the feature of the DNA sequence to the flexibility of the DNA sequence. But what actually is the sequence that are flexible? So in order to find out the feature of the flexible sequence, again, we use this single-strand DNA pool. We actually found that the region immediately 5' to the AGCT motif is the region which has the most effect on the mutability of AGCT. And here we have introduced a pool of single-stranded DNA and look at the different sequences of the from this pool, which are highly preferred by AID compared to those that are least preferred by AID. And when we compared these mutability of these AGCT, we found that sequences that AID most preferred actually are more flexible, which correlates with the dinucleobase stacking free energy of these motifs. And they actually correlate with the mutability of the AGCT that is next to it. And if you look at the motifs, dipyrimidine pyrimidine motifs such as CC, TT, CT, TC, these are motifs that are more correlated to high mutability. So here we were able to find that the pyrimidine motif promotes AID deamination. So if you look at the sequences of the antibody genes, we know that the amino acid of the antibody is selected. For example, in the CDRs, it is enriched for tyrosines or serines. And if you look at the serines, there are actually many codons that code for serines. And the AGC and AGT codon, they are more highly preferred in the CDRs because they are WRC motif. And this is what people have shown to be a codon bias, enrichment of the WRC codons in the CDRs. So here we look at also the DNA sequence in the triplets. And what we found was that besides TAC, there is also enrichment of TAC-related motifs, such as ACT, CTA motifs, which actually correlate to high mutability when they are located 5' immediately upstream of the AGCT. And so this correlates to the flexible sequence as well. If you look at the human J6 segment, there are actually a stretch of TAC repeats there, about 6 repeats there. And we actually wonder, whether there's function of these sequence. So if you look at if you put this sequence into a region into the framework region where normally the AGCT is cold, we now see that if you put in a sequence, the AGCT could be targeted for mutation. So this experiment shows that by synthetically design the sequence, we can unleash AID also at the framework regions. And we think that it can pave the way for synthetic design of a humanized animal model for discovery of antibodies, such as rare antibodies that have mutations even in the framework region. So in this work that I've talked about, we actually solve the question that was first observed by David Baltimore and Leroy Hood. And we show that there is a predisposition of mutations in the CDR. And the mechanism for this is because the CDR sequence have flexible pyrimidine-pyrimidine motifs, which attracts AID to promote hypermutation. And this work was published in Cell in 2023. And this figure was actually a nice figure by David Schatz, which was preview published in Cell Research. So in this work, we think that the coding sequence also plays a non-coding role via DNA mechanics. In this case, in the antibody coding sequence, the flexible sequence actually are used to promote hypermutation. So what is interesting is also when we look at the species that uses a somatic hypermutation to diversify their antibody genes, there is an evolutionary conserved CDR hypermutability. So this experiment was performed using the high throughput biochemistry. So we use the 27 species of the antibody, the antibody genes of the 27 species that are available at the IMGT website. And what we found was that some species that uses the gut-associated lymphoid tissue, such as the cattle and the chicken, they have less of a CDR hypermutability. And these species actually uses gene conversion, which use a pseudogene to replace the variable region exon to diversify their antibodies. And bony fish and cartilaginous fish, they have less mutations as well. So here we think that it's very interesting that the immunoglobulin genes would actually be conserved, and the CDR hypermutability is actually used in species that uses somatic hypermutation to diversify their repertoire. So we also have a review about this in Trends in Immunology. So now we know actually a lot about the mechanisms that diversify antibodies. So are there anything else that are still unanswered? Yes, I think there are still a lot of things that we do not know. For example, in antibody, in anti-HIV broadly neutralizing antibody generation, we know that it still remains a challenge. So in back in 2010, when people have been able to isolate the antibodies against HIV from HIV-infected patients, they found that these broadly neutralizing antibodies, they have very specific, unique characteristics. For example, they have extreme somatic hypermutation, they have frequent deletions and insertions, long CDR3, and these antibodies are also somewhat auto or polyreactive. And we know that anti-HIV broadly neutralizing antibodies, they are generated after many years of infection and through the virus antibody arms race. And mostly patients who were able to develop broadly neutralizing antibodies took actually more than 10 years to generate these broadly neutralizing antibodies. So it remains a challenge for HIV vaccine to be able to elicit this kind of special or rare antibodies. So our question is, first we'll look at the deletions and insertions and long CDR3 And then I'm going to come back to the extreme somatic hypermutation later. So if you look at the broadly neutralizing antibodies that have been isolated, some of them, actually a lot of them, have deletions and insertions in their heavy chain or light chain, which are actually functional for the neutralizing activity. But the mechanism that generate these insertions and deletions are still not known. And some of these antibodies have long CDR3, which allows them to invade the glycan shield of the HIV. But the source of these long CDR3, whether they are generated by RAG during VDJ recombination or AID during somatic hypermutation, is still unclear. If you look at past genetic studies, we know that through knockout of different DNA repairs, AID-initiated lesions can be converted into different mutational outcomes. For example, double-strand break, C to G transversion mutations, or AT mutations through different pathways. And here, from 1998 to 2023, actually many labs have shown that different DNA repairs could generate these different outcomes. But from this work, only 96 indels were obtained. So the mechanisms that generate indels are still unknown at the time when we started this project. So this project, to dissect the mechanism of indels, here again we make use of the passenger allele mouse model because it allows us to look at a single sequence in a high throughput manner. But this time we breed it with the different DNA repair knockout factors. Here we have used about 15 knockout models. And we use an optimized immunization technique to allow us to acquire high amounts of germinal center B cells. And using ultra deep sequencing, again, this is a technology that allows us to sequence the germinal center B cells in an ultra deep way. And we use bioinformatics algorithm to look at the indels. So if you look at the insertions, the passenger allele would have more than the productive allele, although the level is very, very low. 0.31% of the breeds have insertions, but we were able to obtain up to millions of insertions and deletions in this study to allow us to dissect the mechanism. So first of all, if you look at the insertions and deletions, we could actually differentiate them into two different categories. One, which is beneficial, potentially beneficial for the antibody function. And this would require the indels to have 3 nucleotides or count of 3s so that it does not change the reading frame of the antibody genes. And these are common in broadly neutralizing antibodies. And the other is judgmental indels, and these are indels that are 1 base pair or 2 base pair, not in the count of 3. This would lead to a change in the reading frame and would cause the B cell to die because the B cell receptor would have no function. So if you look at the indels that we captured using the passenger allele mouse model, we actually found that the 1 base pair indel events are prevalent events. And only about 20% of the events are in-frame events, which is actually less than what we would predict if 1/3 of the events would be productive. And these experiments also shows us that there could be a difference of mechanisms that generate 1 base pair and the longer than 1 base pair events. If you look at the duplications, insertions, well, first of all, we look at the insertions and we found that the insertions are mainly duplications of adjacent sequence, and this is calculated by the similarity score. most of the insertions are duplications. And what we found was that very rare mutation events actually do lengthen the CDR3 via duplication insertions. This is achievable because our passenger allele has a fixed CDR3 sequence. So if we use a wild-type mouse, we would not be able to analyze this because the CDR3 sequence would be heterogeneous. So we found that these duplication insertions, they can lengthen CDR3. And it's very similar to some of the HIV broadly neutralizing antibodies that have been isolated, which also shows a duplication sequence. So our studies actually shows that AID-initiated insertions can contribute to long CDR3 in HIV broadly neutralizing antibodies. And when we do the dissection of the mechanisms in the different DNA repair knockout factors, each of them carrying the passenger allele, what we found was that actually different mechanisms generate different sizes of the indels. So here again, we can categorize the indels into either one base pair or longer events. And what we found was that the longer indel events actually do require DNA double-strand break because 53BP1 is required for these long indel events. We also found that non-homologous end joining factors, XLF, is required for long indels, for long insertions, and also DNA polymerase lambda is required to generate these long insertions, which could contribute to long CDR3. So in this work, we have knocked out different factors labeled in green here. And what we have shown is that in addition to what is known about how the DNA repair pathways generate the different outcomes, we have added to this chart which DNA repair pathways generate the different sizes of indels. Here, we actually also show that indels at repetitive regions, as short as 2 repeats, is actually mediated by, is actually suppressed by the mismatch repair pathway. And this work was done in collaboration with Pan Hammerstrom and Roberto Ciali, and we published this in Science Immunology in 2023. And we think that the significance of this is that in the future, if we want to do humanized antibody mouse models to select for rare or broadly neutralizing antibodies, we could probably manipulate these factors to allow for higher opportunities to generate antibodies that have longer indels for selection. So this is the work that shows probably we can, in the future, do synthetic manipulation on the trans factors. Okay, so I'm going to go in the last bit to talk about our unpublished work. And this is work that is still on the HIV broadly neutralizing antibodies subject. And in this work, we are looking at the extreme somatic hypermutation and what causes this extreme somatic hypermutation. So when we look at the antibody sequence from HIV and from other antiviral antibodies, we know that these HIV antibodies have high amino acid mutation frequency. But not only that, it also have high levels of mutations that require multiple base substitutions. So here on the left, I've shown a table that shows, for example, a DBS, a double base substitution mutation, or single skip by one substitution, or triple base substitution. So these are changes in the codon mutation, which altogether we call it multiple base substitution. So if you look at this VRC01, this is a multiple base substitution plot. We can see that it has a lot of mutations, not just in the CDRs, but also in the framework regions. And furthermore, there are a lot of mutations that involves multiple base substitutions, such as double base substitutions and triple base substitutions. So the mechanism of that generate these multiple base substitution is not really known, and we were thinking of using our passenger allele mouse model to study these mechanisms. So in this work, unpublished work, we have generated passenger allele mouse that carries either the human VH1-2, 330, or 459, which are commonly used in HIV broadly neutralizing antibodies. So the way we analyze the multiple base substitution is very similar to previous studies. We immunize these mice for 9 days to obtain their germinal center B cells and then perform a deep-seq downstream analysis. So what we found was that even in early activated B cells, we can capture double base substitutions in these mice. And this frequency is actually higher than deletions, insertions. But for triple base substitutions at these early stages is still quite rare. So we then look where the double base substitutions are. So this is a profile, somatic mutation profile of the single base substitution. The double base substitution profile is actually very similar to the single base substitution, except that we do see some peaks at these motifs like CC and GG. And this is the mutation spectrum, the double base mutation spectrum, according to the COSMIC from the Sanger Institute. We found that mutations like CC to TT, they are more prevalent compared to other types of double base substitution. If you look at how the amino acid will change from these multiple base substitutions, so for each position in the antibody gene, we could actually check how each of these amino acids change to the other 19 amino acid. And what we found was that for each of these amino acid labeled in gray, this is amino acid that require only single base substitutions. They are more frequently generated compared to mutations that require double base substitutions, which is in blue, or triple base substitutions, which are sometimes not found in our study, which is in red under this black line. So here we show that mutations that require multiple base substitutions, they are actually very rare, and they are 1,000-fold less frequent than single base substitutions mutations. So the multiple base substitution mutation is the bottleneck to generate these broadly neutralizing antibodies. And with our intrinsic mutation data, now we are able to know, for example, the simplest mutation trajectory for each codon. For example, if we want to have the F amino acid from a germline K, it can go through many routes, but the best or the easiest or simplest route for it to mutate to become F is to go through this path in bold. So this is something that we can get from our data. And if you look at the different codons here from the 3 passenger alleles, now we know for each of these codons the exact mutation frequency for it to become an amino acid that requires single base substitution, and also an amino acid that requires double base substitution or triple base substitution. So what I want to say here is that in an unselected condition, in a physiological condition, the mutation trajectory for an antibody is fixed. And with this, right now at this stage, we only have at the codon levels. So I think in the future with AI, and also with more data, we would be able to have the intrinsic mutation trajectories for all heavy chain variable region and also light chain variable region. And this would provide a reference map for the analysis of B-cell repertoire. So in the future, I think given any B-cell repertoire, we would be able to identify mutations that are intrinsic, which means that it is usually generated by AID, regardless of whether there is selection or not. And also we would be able to identify mutations that are selected because we know intrinsically it happens very rarely. So the implication of this work is, so for HIV vaccine, what has been proposed by Bart Haynes and John Mascola back in 2013 was that in order to guide the broadly neutralizing antibody maturation, we could design different immunogen at different stages to activate either the germline or the intermediate B-cell clones. So based on our study analysis, we now know which pathway is the easiest for an amino acid to mutate to another amino acid that require multiple base substitution. So perhaps we could use this knowledge to design immunogen that can specifically activate the best single base substitution B-cell clone, for example. And also, in the future, we would try to understand how the mutation trajectory would result in broadly neutralizing antibody activity or autoreactivity. So if you look at this HIV broadly neutralizing antibody, BRCA1, it has a lot of mutations, and possibly a lot of these mutations also results in autoreactivity or polyreactivity. If you look at autoantibodies from some of the autoimmune disease patients, some of them also have a lot of mutations. And so the challenge here is how do we generate broadly neutralizing antibodies while avoiding autopolyreactivity? So these are some of the things that I think in the future would be of great interest for us to study. So this is sort of my last slide, which is a summary of our research. So my lab is studying antibody diversification, mainly the molecular mechanisms. And in this aspect, we have looked at the cis factors, which is the sequence of the DNA, and also the trans factors, which is looking at the different DNA repair pathways that generate different mutational outcomes. So for the cis factors, I've talked about the CDR bias hypermutation based on the DNA sequence flexibility, and also the mechanisms that generate indels that could promote the generation of broadly neutralizing antibodies. So one of the thing that I didn't have time to talk about today is our work on humoral immunity, and this was during COVID a few years ago. Our lab found that vaccination with the inactivated virus vaccine can induce immune imprint, and this immune imprint actually was erased or was actually overcome when there is a multiple infection. So these are some of the work that we have published recent years. And our lab has also written several reviews that I've listed here. So finally, I want to thank my lab members and my collaborators. So the work that I've described here have mainly been done in collaboration with great collaborators, Changpan Hammerstrom from Karolinska, Fred Alt, who is my postdoc supervisor, who keep collaborating with us, and Roberto Cioli from Harvard and Toronto University, Fei Longming at Shanghai Institute of Biochemistry and Cell Biology, and Leng Siew Yeap, Fubin Li, Chuan-Xing Huang, who are my colleagues at Shanghai Institute of Immunology, Xiaoqi Zeng, who is a mathematician, Lin Taida, who is a molecular stimulation dynamics expert, and Lei Ye, who is a clinician at the Region Hospital Shanghai. And the work that I've talked about today was performed by my students and postdoc. The work the work on CDR hypermutability was performed by Yan Wang. The work on indel was performed by Chen Hao, Chuanzhong Zhan, and the work on multiple base substitution was performed by Chaoyang Yan. So thank you very much for this opportunity to give this presentation. If there's any questions, please welcome to contact me at my email address. And also, I think there's a link at the end of this presentation here. Thank you very much.
Great, thank you. Thank you for a wonderful talk. And as mentioned earlier, and also show on this slide, it's the current tradition for global immunotalks. We'll have a Q&A session on YouTube. So you will scan this barcode, QR code, and leave your comments on this week's recorded video for Dr. Yeap. And then she will answer that in due course. And so thank you for your attention. I think that's all for today's Global Immunotalks. We'll see you next week.